Good afternoon. My name is Michelle Gorder, and I am the Supervisory District Conservationist with Montana NRCS in the Bakerfield office. This presentation is a condensed version of a 3-year high stock density grazing project that has been ongoing with producers and NRCS in Fallon County, Montana. I'll be going through our data collection process in the field, some of our results, and a brief overview of the annual grazing plan development. I would like to give a brief introduction to our other presenters that will be joining in later to address the soil microbial aspect of this project and implementing the plan from a producer's perspective. Serafina Clays is our in-house resident expert on soil microbiology, previously employed in the Bakerfield office through a grant from Montana Association of Conservation Districts. She is now an independent soil microbiology consultant. Dave Hayden is a third-generation rancher on the Hayden Ranch, 30 miles south of Baker. Thank you for our partners at MACD and Montana GLCI for their support in this project. This project was located in Fallon County in southeastern Montana. We worked with 4 producers on 5 project sites, 2 crested wheatgrass and 3 smooth brome. Fallon County is in a 10 to 14 inch precip zone. The overarching goal of this project was to evaluate the use of livestock as a more economical means of rejuvenating pastures and improving soil health. This would be accomplished through hoof action from livestock, which would incorporate the plant residues and manure into the soil and therefore stimulate microbial populations, improving nutrient cycling and plant health. We expected to see that with improved soil health, we would start to see increased plant diversity, improved plant vigor, increased biomass and water infiltration. We also expected to see a reduction in percent bare ground and hope to see some changes in the plant community with stimulating the latent seed bank. The focus of this project was narrowed down to improving plant condition on tame pastures within set stock density parameters. Producers had to be willing to graze at a stock density between 100 and 250,000 pounds of livestock per acre for 3 years in a selected area. Most selected sites were around 40 acres, with the largest being 122 acres. Time commitment for grazing at this level of intensity varied based on the herd size that they were working with, their set goals for stock density, and the forage production for the year. Permanent monitoring transects were established at each site with GPS points at the 0 and 100-foot ends of the transect. All data was collected on or around the transect. Samples were collected as shown on the slide. Infiltration tests at either end. Soil was collected in a composite sample and divided 3 ways for the separate analysis. Biomass data was collected at 25 feet off either side of the transect and moved incrementally each year sampled to maintain the integrity of the line point transect. The following data was collected: line point, monitoring photos, forage production, infiltration, penetrometer readings, and temperature. Plants were clipped and sent in for tissue sample analysis, and composite soil samples were collected and sent off 3 ways for specific testing. Serafina will explain a little more on some of the testing that was done.
So the soil microbial communities that are often broken down into their functional groups, we have bacteria, fungal, protozoa, and nematodes. All 4 of these groups have an interesting interaction with one another, and especially plants. Throughout the course of this project, there were 3 main tests that we utilized to track the microbial communities. These communities are always fluctuating, and many factors play into their survival and their ability to thrive. So as far as biological testing is concerned, we did 3 main tests. The first one was the PLFA test that's conducted at Ward Labs. And this test analyzes the phospholipid fatty acids, or the PLFA. The test gives a representation of the soil microbial biomass, and then it identifies the presence or absence of various functional groups based on biomarkers. The next biological test was conducted by Mr. DNA of Shallow Water, Texas. And this is a DNA sequencing lab that extracts the DNA of all of the eukaryotic and prokaryotic cells in the soil sample that we sent. Each report is very extensive and it gives an entire taxonomic report all the way down to species. So with all of this extensive information, we look forward to a university student picking it up and eventually running it through an analytical program to try to find some correlations. The final biological test that was performed was an in-house microscopic evaluation of the soil sample, and this was performed by myself. The samples are evaluated fresh for quantity, viability, and diversity within each microbial functional group. This gives a real-time view of what's going on and allows us to take pictures of the microbes for documentation and then later for comparison year to year. These results are calculated into either a number per gram of soil or a microgram per gram, depending on the functional group. This evaluation usually takes about an hour per sample. So this slide indicates samples on both ends of the microbial biomass spectrum for a microscopic view, from a microscopic view. Neither of these pictures are from this project, but they're here to show the drastic variations that can be there. So on the left, we have a depleted soil where only bacteria is viewed. And then on the right, we have this diverse soil, which is an old growth forest. And we can see all the functional groups in this one field of view. So plants and microbes, they have this symbiotic relationship, and the soil microbial life plays a critical role in plant health. Microbes in the soil facilitate nutrient cycling and release nutrients to plants through the breakdown of organic matter. The plants in return then use an astonishing amount of their energy to release sugars through the roots to feed these microbes. Studies on plant and microbial interaction is always advancing, and this field has some pretty exciting information that's developing. Soils that are supporting a healthy microbial community, well, they have this capacity to convert the complex organic matter residue and then make these nutrients available to plants, specifically nitrogen and phosphorus. So we can have these free-living nitrogen-fixing bacteria that also play a huge role in converting nitrogen into a plant-usable form. So having this team of beneficials, it can also help prevent disease infestation. It can help outcompete pathogens that may be present. It can produce plant hormones, which will then stimulate root development, and ultimately this affects the plant growth and health. So as far as soil structure is concerned and microbes, well, they're the builders, and they're producing these organic acids and glomalin that aggregate soil particles and then improve the infiltration of water and oxygen. So the fungal community, that usually takes the spotlight, and it does in this case too, for good reason. For the sake of this presentation, we're looking specifically at our fungal biomass. There are so many fungal species that benefit the health of our soil, and very generally speaking, they can extend the root system of plants and they facilitate nutrient uptake. This increase in surface area of plant roots, it ultimately gives plants an advantage during times of drought and stress. And microscopically speaking, the fungal population on all the sites increased at the conclusion of this project. A notable increase in the hyphae cell wall width was observed on sites A, E, and D. The cell wall width, that can be observed as kind of an indicator of the age of the hyphae, because as the fungi age, the wall and the septa thickens over time with chitin and glucan. On these three sites, I did notice an increase in the thickness of both the hyphae cell wall and the septa. So we'll conclude with this graph that's from the PLFA results conducted by Ward Labs. And it's depicting the changes of the PLFA total microbial biomass, which is the graph on the left. and the fungal biomass on the right over the course of the project. And knowing that the microbial communities are always fluctuating, especially in our brittle environment, we must monitor trends over time. So when Michelle goes over the weather events in the upcoming slide, keep these line graphs in mind to see the correlation in fungal biomass that kind of follows the precipitation. So the fungal biomass, for instance, among its ups and downs throughout the project, it's still overall trending upward.
Weather events factored into the overall results of the project. In 2021, Fallon County experienced a significant drought, and in 2022, we experienced two back-to-back spring blizzards in April. From 21 through 23 and into this summer, our area saw significant outbreaks of grasshoppers. The precipitation graph is showing total accumulation from September the year prior to June the year that the data was collected. You'll be able to pick up on some of the impacts of these weather events in the next few slides. Data was collected at each site in early June prior to grazing. Biomass was clipped in two locations, 25 feet out from either side of the permanent transect. The biggest jump that we saw in production was on Site C, which was primarily a smooth brome field. Management prior was hayland with aftermath grazing, and production on this site went from 640 pounds in 2021 to 3,458 pounds in 2023. The trend in organic matter has been interesting, and other metrics being evaluated follow similar ups and downs. Data on the slide shows measurements each year, 2021 through 2024, and the percent change in italics is from 21 to 23. There was a dramatic increase in organic matter percent across all sites initially, ranging from 0.5 to 1.2% increase, followed by a declining trend. And the biggest jump in organic matter was seen on Site E, which was smooth brome, at a 1.2% increase. This field had previously been in season-long grazing. Overall, stock density across the project sites ranged from 105 to 250,000 pounds of livestock per acre. The biggest change in stock density is Site E, that went from a 240-acre pasture prior to the study to 0.37 acre temporary paddocks during the project, from a grazing period of 70 days prior to and moving 7 times per day during the study. Stock density on this site went from 309 pounds of livestock per acre prior to the study to 168,593 pounds of livestock per acre in 2022. We had one participant that elected to stock at the higher threshold of 250,000 pounds per acre of livestock throughout each year of the study. Plant and residue cover indicate a slight decrease on most sites from 21 to 24. There appears to be a trend being observed between stock density, rest interval, and infiltration rates. There was an observed decline in infiltration rates, with a noticeable compaction layer at 1 inch across all sites in 2023. That apparent compaction layer has been resolved on most sites in 2024. Ideally, the sites would be rested a full 12 months between grazing events, but through the drought, Site D elected to defer grazing in 2021, and Sites A and B intended to defer grazing, but went ahead and grazed in the fall of 2022. Aside from the data, some of the more interesting aspects of this project were observations in the field. Positive changes in soil color and structure were observed on most sites. The most apparent change in soil structure and color was on Site C, which correlated with the data coming back in soil organic matter. We had hoped that by the end of this project, we would start to see more native species showing up on the sites, with the idea that high stock density and short duration would help to stimulate the latent seed bank. During our field data collection this year, on Site B, there was an abundance of needle and thread grass observed, and on Site D, there was an abundance of green needlegrass. These changes that were obvious looking across the pasture didn't translate to any significant changes recorded in the line point transect, but it was a change that was noticeable in the field. In this section, we'll go through general grazing plan development and what that looked like from the producer's perspective. Forage was clipped prior to grazing each year, depending on the herd size and target stock density goals. Plans varied between 100 and 250,000 pounds of livestock per acre. Producers moved between 3 and 12 times a day. Temporary paddocks for the moves ranged from 0.37 acres to 2.9 acres. And total grazing time ranged from 2 days during the drought to 22 days in 2022 after the spring blizzards. This spreadsheet is one of the tools that we use when developing the layout for the grazing plan each year. We input data for class of animal, average weight, and the percent dry matter intake, field area in acres, field width, dry matter, or the actual forage clipped in the field, utilization rate targets, and moves per day. Plugging in the daily moves will both get us in the range of where we need to be for stock density and will calculate the width of the paddock. I used these paddock dimensions to come up with a layout for the grazing plan that I would give to the producer, along with the moves per day needed to meet the herd's forage needs. In the next few slides, Dave Hayden will take us through putting the plan in place from a producer's perspective.
This pasture is 122 acres divided into multiple sections for high stock density. The outside border in black is permanent fence. All the interior blue lines are temporary electric polywire. The blue dots represent the water. To lay out the fence, the first year we used GPS. Second year we used a pasture map. The third year we used the drone, which worked a lot better. It was a lot easier because when starting the drone, it shows on the monitor ground zero, and when you move forward, it measures in feet your distance. This made it very easy to measure out the pasture on the whole fence. There was a hill in the middle of the field that kept us from seeing the fence on the other side. So using a drone made it easy to see over the hills and find the flag that were placed that we needed to connect to. The drone saved time, wear, and labor on equipment. We set up the top of the section first, started the cattle grazing in the middle where the water tank is, and then grazed to the left until they hit the permanent fence. Then they went back to the water tank and grazed to the right. There was no need to set up back fence because the cattle were headed to fresh pasture with each move. The rest of the fence was set up while the cattle were grazing. There were 12 moves per day, moving every hour. The short polywire runs are threaded through the ring of a step-in post, and a short piece of rope on the end that attaches the insulator, then is pinned to the ground with that stepping post. This way you can handle the wire without turning off the charger. So the long runs are the wire that's hot, and when you run that short one through that ring, then it causes it to be hot. Rolling up the wire was quite a task until we come up with a wire winder and made it a lot faster. The first 2 years we used yearlings. The last year we used cow-calf pairs. The yearlings were much quieter and waited till we pulled out the fence for the next move. The cows were impatient, did a lot of bawling for their calves, not that they were hungry. This became a habit. One water was set up between row 2 and 3, and when those rows were finished, they leapfrogged to row 3 and 4. The water system was supplied by an above-ground heavy hose connected to an underground water source 1/2 mile away. The poly tank and the drinker hold 3,400 gallons. When the poly tank is full, it puts out 33 gallons a minute to the drinker, and when it's half full, it's still 26 gallons a minute. The high flow rate keeps livestock from standing around waiting for water. In this photo, you can see how we moved away from the water as we took out the poly fence from the paddock, and the cattle are moving into a fresh one. Next time we would stockpile forage for 12 months before doing a high stock density project. This is important when trying to get cover and carbon on the ground and using cattle as a tool. This high stock density project made a great impact on our soil health in only 3 years. Anytime you can shorten up your grazing time and lengthen your rest time in a pasture is very beneficial. We've been doing holistic management for 20 plus years. We currently use just half of our place in 1 year. The other half is rested for 12 to 18 months, having recovery time that is very important.
So that's a condensed version of our project. And in addition to this, Montana NRCS has filmed the high stock density project on site at the Hayden Ranch. They filmed in June of 2020 and has since went on to publish their video on their Montana NRCS success stories. The QR code on this screen will take you to a video of the grazing project in action. Here's our contact information, and we thank you for your time.